Today we are looking at the nervous system, in particular the structure of a neuron and the generation of an action potential. Now, cast your minds back to your IG studies and you'll recall that the central nervous system is formed from the brain and spinal cord, and there is of course a peripheral nervous system, the nerves and ganglion extending from the spinal cord. Cast your mind back, also recall there are three types of neuron: the sensory, the relay and the motor nerve. Notice the positions of the cell bodies to distinguish the three types of nerve. Please recall the parts of the nerve, specifically the nodes of Ranvier and the myelin sheath. Now, we have always called these nerves the sensory, the relay and the motor neuron. There are actually, at AS and A2 level, different names for these. The sensory neuron is also called the afferent neuron. The relay neuron can be called the interneuron, like an intermission between the two. And the motor neuron is now called the efferent neuron. These are the new names you can use, but they are at the moment interchangeable. So how do we generate this action potential? Well, it is a reversal of a potential difference across a membrane. So what we need to do is we need to move ions either against their concentration gradient or rapidly allow them to flow from their concentration gradient. It is the flow of a charge, but not as you are thinking now. To create this potential difference, we need to cast our minds back to AS. We need to recall some of the mechanisms which allow transport across the partially membrane, specifically the voltage-gated channels and the sodium-potassium pump. The ligand-gated channels will become relevant in later videos. With regard to the sodium-potassium pump, remember that 3 sodium ions are pumped out for every 2 potassium ions pumped in. This will start to generate a potential difference across any membrane where these pumps are working. The first thing we must establish before creating our action potential or sending our nerve impulse is the resting potential. This is achieved by using these sodium-potassium pumps. As I've already mentioned, sodium is pumped out in 3s and potassium is pumped in 2s. Automatically, this creates a +1 relative charge to the external fluid. The sodium that has been pumped out has no open channels to allow it to leak back in, but the potassium does possess these leak channels, so it can flow out of the axoplasm. This further pushes the charge on the extracellular fluid up, pushing our relative inside to a more negative position. Once this has reached a negative -70, we call this the resting potential. Sodium is pumped out and cannot re-enter. Potassium is pumped in but then leaks out to create this additional relative charge. Now, this diagram summarizes the action potential or the generation of a nerve impulse. It is a little bit of an overwhelming diagram, so we will take it piece by piece. Here you see a kind of almost heartbeat-like diagram showing a movement of charge or an impulse. This is, as the diagram describes, the resting potential to the action potential, the generation of an impulse. The threshold point, labeled 2, shows us the point at which the impulse will be propagated or spread. Any impulse below this, -50, -55, will not generate an actual nerve impulse. As you see in diagram 1, the resting potential has been established. As you've already looked at, this is about a -70 in most textbooks, maybe -60 in some. It is a condition where the inside is negative relative to the outside. If a stimulus is received, it may start to open some of these sodium voltage-gated channels. A few open, -70, -60, -58. If nothing else happens, these channels will close, the sodium-potassium pump will work, and we will return to our -70 resting potential. We have not overcome threshold. However, if a stimulus is received, number 2, we see a rapid opening of not 1, not 2, but many sodium channels. This causes an influx of sodium, pushing our negative rapidly to a positive. This is the depolarizing wave of the action potential, shown as 3. Once this has reached its peak, the sodium gates close or the channels are inactivated, but potassium channels are still open. This permits potassium to efflux, to leave, to go out of the axoplasm. This will become more negative. We are now dropping from plus 40, plus 30, plus 20, heading back towards our resting potential. Now, this whole process is extremely rapid. Therefore, a sudden change in these sodium channels closing, potassium channels opening, will cause what's termed hyperpolarization. We will undershoot for our minus 70. The sodium channels will close, the sodium potassium pump will do its job, and we will return to minus 70, resting potential. Another stimulus would trigger another action potential. The impulse will be propagated. These two graphs summarize what the previous slide was showing us. Note the sodium influx corresponding to the depolarizing wave, and once these sodium channels close and the potassium channels slowly open, we see the repolarization, the sudden drop. This happens extremely rapidly, so there is hyperpolarization. Once the potassium channels close, the sodium potassium pump returns to its normal function and we re-establish the resting potential. This final slide shows the full nerve impulse from the initial resting potential right through the depolarization, repolarization, hyperpolarization, and finally the re-establishment of the resting potential. I will leave it here for a few seconds for your review.